In this video, we will discuss about how this fatty acid mechanisms in the cancer cell works and how it will lead to the how it will help the cancer cells in in its progression and also the spreading of the cancers across different part of the body, that is metastasis of the cancer. So we will start with this fatty acid mechanism in the cancer cells. So in the cancer cells, because of the mutations, what happens is that the CD33 receptors has been translocated to the receptors, and where it will act as a lipid intake mechanism, where it helps in the endocytosis of the lipids within the cells. And when the lipids are entered within the cells, then they will be converted into this lipid droplet with the help of this DGAT1, or either they are completely converted into lipid droplets, or they are being utilized through by this beta oxidation process to convert it into the energy form. And the another way by which the energy is utilized in this fatty acid mechanism is through this glucose intake with the help of this GLUT1, where glucose is converted into the pyruvate, and thereafter this pyruvate and this acetyl CoA beta oxidation and TCA cycles in combined helps in this lipid formations uh, with the help of several enzymes such as ACL1, ACC, FASN, SCD1, that will helps in the increase in the lipid pool within the cancerous cells. And however, so this is about the fatty acid mechanism in the cancerous cells and how they helps the cancerous cells in uh, proliferation and metastasis. Is that the enzymes, this MAG1, enzymes helps in modifying the tumor microenvironment with the help of lipids, by utilizing the lipids. And during these mutations that occur within the cells, there are some de novo lipogenesis methods also occurs, will also increase the lipid pool. And this and the raw materials for this MAGL is the lipid pools which are already present in the excess amount in the cancerous cells. So it is easy for this MAGL to work towards modifying the tumor microenvironment in a way that it will not only help the cancer cells in uh, immune evasion, but also helps them in the in its proliferation characteristics and metastasis properties. And so this is about the fatty acid mechanism. And what are the considering this knowledge, what are there are some strategies that have been developed to target this lipid uh, to treat the cancers, and those are known as lipid targeting anti-cancer approaches. And some of the those approaches are mentioned in this figure. For example, this p10 gene is responsible, has some immune tumor suppression properties, and this is usually inhibited within the tumor cells, or we can say this in mutated, or due to epigenetic changes, the p10s are not able to produce in the cancerous cells in efficient amount. So by targeting this p10, by upregulating the p10s, they can increase the immune response. They can increase the immune suppressor properties within those cells. At the same time, they will control the. It will also helps in controlling the uncontrolled growth rate of the tumor cells, or in general terms, we can say that it will suppress the tumor cells growth rate. And simultaneously, the RB1 gene also has the similar properties of immune suppression. It will helps in the upregulations of this RB1, helps in the controlling of the growth rate, or cell divisions rate within the cancerous cells. The TP53, we have discussed this TP53 properties many times. Uh, it has the functions to decide. It has a function to decide whether the cell should go for its repair, DNA damage repair, or cell division, or it should go for this cell death. How and this all these three genes are suppressed within the tumor cells, and these genes can be targeted as they are not only related to the cell control growth rate, but it will also they are also related in this lipid acid formations. So by targeting or by upregulating these genes, they will have the double impact, that is reduce the lipid formations within the cancerous cells, that will reduce the tumor microenvironment effect. At the same time, they will also reduce the proliferation or uncontrolled growth rate characteristics of the cancerous cells. So this is how these lipid targeting anti-cancer approaches are thought to work in the theoretical manner. So I hope you got a basic idea about this fatty acid mechanism in cancer cells, and about also some insight about this lipid targeting anti-cancer approaches. If you need to understand more about this topic in detail, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.